Hi, my name is Rachel Moss, and I'm a third-year PhD student in the Bioinformatics and Computational Biology program at the University of Minnesota. Today I'm going to be talking about our project that explores the role of genetic ancestry in EWS-FLI1 induced epigenetic and transcriptional programs in Ewing sarcoma tumorigenesis. For a brief background, Ewing sarcoma is a rare but deadly bone and soft tissue tumor that's found primarily in children and young adults. These sarcomas consist of small round blue cells with prominent nuclei, with just over 400 cases diagnosed each year in the United States alone. Overall survival has actually remained around 60% for decades now and drops to around 35% for those that present initially with metastasis. So what we know about Ewing sarcoma is that it has a dominant genetic driver that's caused by a chromosomal translocation, and this generates a fusion between the EWSR1 gene and members of the ETS transcription factors family, with the most common form being EWS-FLI1. EWS-FLI1 then binds to GGAA repeats in DNA and creates novel enhancers that turn on oncogenes, while simultaneously repressing genes with the single GGA motif. EWS-FLI1 therefore acts as an aberrant transcription factor with pronounced changes to the transcriptional landscape. So studies like this one by the Ewing Sarcoma Cell Line Atlas find that knocking down EWS-FLI1 in cell lines produces thousands of changes in gene expression. But interestingly, EWS-FLI1 is notably toxic to most fully differentiated cell types. But if you add it to a multipotent cell type, like a mesenchymal stem cell or a neural crest cell, we find that these cells tolerate EWS-FLI1 long enough to have potential as a model. It also implies a strong developmental context to Ewing sarcoma tumorigenesis. It's well established that the biggest risk factor for Ewing sarcoma is ancestry, with a more than 9-fold difference in incidence of those primarily European ancestry compared to those with primarily African ancestry. Although there's an established polygenic architecture to Ewing sarcoma risk, the underlying allele frequencies by population do not fully explain the differences in incidence. We believe that polymorphic GGAA repeat length likely underlies most of these risk loci in the GWAS data and could be a key to discovering novel loci. Our hypothesis is that genetic ancestry will dictate the molecular and functional responses to EWS-FLI1 expression, with permissive European and impermissive African genomes. But because there are so few naturally occurring cases, we actually can't study African genomes in vivo. At the same time, the cell state must be receptive to EWS-FLI1. Therefore, we've created this induced pluripotent stem cell model in which we can identify donors based on their degree of admixture. To build a model that includes all aspects of permissive genetics, we obtained several iPSC lines from donors with varying ancestry from 100% European to 100% African. In order to create the permissive cell state, we differentiate to neural crest cells. We next add the EWS-FLI1 oncoprotein, which is transduced with a lentiviral expressing either a GFP reporter for control or a GFP with EWS-FLI1 in the cassette. Finally, we measure cell survival by using the GFP reporter via flow cytometry, gene expression through RNA-seq, EWS-FLI1 binding to DNA using cut and tag, and local ancestry inference using RFMix. So, in fact, we were not only able to show that the iPSC-derived NCC stably expressed EWS-FLI1 for a long enough time to do our downstream experiments, but when we measure dropout of the cells within a given population admixture level based on GFP, there are clear differences in survival by degree of African ancestry, with higher African ancestry declining faster than those with higher European ancestry. Next, we wanted to look at global expression patterns in response to EWS-FLI1 and establish similarity to other Ewing sarcoma studies. This volcano plot shows gene expression changes at 96 hours post EWS-FLI1 induction using all cell lines. And we see those large-scale gene expression pattern changes that we were expecting, with around 9,000 differentially expressed genes. And these differentially expressed genes additionally are enriched for established Ewing sarcoma expression patterns when we do gene set enrichment analysis. After establishing the similarity of our model to other Ewing studies, we proceeded to look at the differential expression that corresponds to degree of admixture. We identified about 3,000 genes that are differentially expressed based on the degree of admixture. Similarly, we looked at EWS-FLI1 occupancy in the genome that has differential binding based on percentage of African ancestry. Out of a total of 3,000 loci with EWS-FLI1 peaks, over 600 of these actually show differential binding in relation to percentage of African ancestry, and that maps to about 300 unique genes. We next asked, which of these genes are both differentially expressed and differentially bound based on ancestry? And we identified 80 targets that are potential ancestry-linked differential EWS-FLI1 response, or as we call them, alder loci, that could have a critical role in the early transformation of cells in Ewing sarcoma. In conclusion, we have a novel, scalable iPSC model of early EWS-FLI1 response that allows us to look at even impermissible genomes. We have found thousands of places that are differentially expressed or bound in our initial analysis, and we've identified as many as 80 of these alder loci, which we think could be potential targets of therapy. Thank you so much for coming to my virtual poster talk. I appreciate any questions or comments, and I hope you enjoy the conference. Thank you.